I always wanted to pursue my higher studies. So in the same year, I'd actually applied for higher education and grad studies, especially in masters in the US. Um, I had applied for a couple of universities, and then, well, I just picked to go to Florida because of the weather conditions and different things. And plus, I really got an opportunity where in the beginning itself, before I went, I had a really good contact with the head of the department at the university, and I had spoken to him about giving me some kind of like assistantship for research or teaching, which would actually help my stay here for the next 2 years. And so I came to the university in the spring of 2014, and immediately, like the first semester, I started my research. So my research for my master's was entirely different topic, where it was based on analyzing how the chemo drugs would affect people with alopecia. And through that, I have a couple of papers and I think conferences that I've attended and presented the study. If anybody wants any like details on that, and just let me know, email me, and I can let you know about it as well. And once I finished my master's, spoke to my professor or my PI, and he was really interested to give me a chance to do my PhD. And he was ready to like sponsor for the whole thing. So I immediately started after I graduated from my master's. That was in spring 2016. And yeah, it took me about almost 4.5 to 5 years to complete my PhD. So my PhD was, yeah, a really good experience. What I had, I learned a lot of things, and I know compared to our country and here we have more exposure to different, uh, especially with respect to equipments, and obviously with all the money that with respect to research. I don't know how it is now over there with respect to getting funding, but here it's pretty hard. You need to be really good at what you do. But yeah, once you get recognized for it, it's always there. And hopefully that will give you guys some motivation to like keep up with what you're doing. And I know most of you have a lot of questions regarding what you're doing and your interests. And I know I was like that too when I was in undergrad. But trust me, the field you guys are in right now, currently studying, it's really going up. Not only like here where I am right now, but I can tell you in different countries. So you guys are doing great. Keep up the good job. I'm going to continue with my presentation of what I did for my PhD. So don't hesitate to ask questions. Just unmute yourselves and just ask me anything you want in between the slides. So my PhD dissertation was optimizing bioink and 3D printing using this bioink and trying to test the effect of microgravity on the bioprinted constructs which had cells embedded in them. So first of all, to go into bioprinting, I'm not sure how much exposure you all have with respect to bioprinting. So the main need for this whole topic of bioprinting was because currently it's one of the main topics that's being explored here everywhere with respect to our field. And it basically involves mimicking native tissues, especially to achieve vascularization, which is one of the main uh, sources of uh, factors that's been looked into and which is very difficult to achieve to mimic any kind of tissue. And the next thing, just to go about a little bit about model systems. So there have been a lot of research with respect to 2D models with just seeding cells on any kind of surface and studying what the effects are of exposure to anything such as drugs, or in my case, with respect to microgravity. But there hasn't been much studies done on any kind of 3D cultures because it's a current upcoming field. So the importance of having the 3D cultures is it mimics the environment of the where the cells are or the tissues are much better than a 2D surface. So next, coming to microgravity. So microgravity is one of the most significant stress factor that is experienced by astronauts when they go to space. And most of the study which has been conducted, or if you have seen like papers or any kind of like online based things with respect to astronauts in space, a lot of study has been conducted with respect to microgravity and what the astronauts actually go through in space. And some of them are really, really interesting to see, where microgravity actually changes or certain things, especially with respect to our cells and our genes, and it alters like cell signaling, and it changes like the hydrostatic pressure gradient, and it causes mechanical stress, especially on your cells. So another main factor that has been looked into is bone loss, which there has been a lot of research and you can find a lot of things about it online, but there hasn't been a lot of research which is looked into specific organ systems or models in the human body with respect to microgravity. So, like I mentioned before, with respect to 3D printing, the main problem that is currently being addressed is to achieve this vascularization, which is really important to have or mimic the entire tissue. Without achieving this, it's really hard for it to evolve into an actual tissue and to make sure that we have something which mimics the environment to test it for any kind of effects. And there have been, with respect to bioprinting, there has been a lot of different approaches or methods that has been used to achieve this vascularization. I won't go into details, but some of them are scaffold functionalization, where you print scaffolds with channels and then seed cells in these channels in order to mimic the natural environment of these cells and to uh, evolve it into a tissue. Then there are cell-based techniques where you use different cell lines to make this particular achieve this vascularization of tissue. And then you have bioreactors. Then you have MEMS-based approaches, which is related to microfluidic systems. And there's something called modular assembly, which again involves different cell lines. And you use these different cell lines to bioprint and use them to make or 3D bioprint constructs with cells. And next is in vivo systems, where you print these constructs and you can maintain them in the incubator and as a bioreactor as well to achieve this vascularization. Next, coming to bioink. So I don't know how many of you know what a bioink is, but a bioink is a material that is currently being used, or it's a name that is termed for something that contains any kind of biomaterial and it has cells in it. So with just the biomaterial itself cannot be called a bioink, but once you introduce cells into that bioink, into the hydrogel, for example, in my case, it's called a bioink. So it's called a bioink basically because it helps when in printing, where if you seed cells in this particular component, then it helps the cells from not dying out or losing its viability. So there are different kinds of bioink as well, with respect to what application you want to use it for. So they are divided into different categories, such as naturally derived mammalian, for example, gelatin, collagen, and fibrin, which are one of the three main biomaterials that are being used. And next, we have naturally derived non-mammalian, which with respect to alginate, chitosan, and agarose. And then we have synthetic, which is PEG and then pluronic. And then we have hybrid, where you can use two different materials from two different categories and make it a hybrid bioink. And each material has its own advantages and disadvantages. So now coming to what I did for my dissertation. So I basically divided it into three different steps. So the first step involved trying to build a bioprinter in my university. So the bioprinter was, I know we can buy a bioprinter. I mean, it's expensive, but it was kind of like a project for like the seniors here. So what we did was we built a bioprinter from from like scratch, where so the main difference between the bioprinter that we printed and that is there in the market right now is our bioprinter doesn't have to be inside the laminar flow hood all the time, because when we do any kind of biology or cell work or any kind of biology work, it's supposed to maintain sterility. So most of the bioprinters right now in the market don't have that capability. So you need to put the bioprinter into the laminar flow hood to achieve sterility. So what our bioprinter contained is it had its own HEPA filter and exhaust fans. So we could just place it on the desk, and it had a cover up with a lid, which we could just close once we started printing, and it had its own UV light, which we could sterilize it. So we built that printer, and we tested the printer for its printability, for its capability of printing, etc. And actually, the nice part of it is it actually won the senior design award from Northrop Grumman for the best engineering or biomedical related product. And so then the next step was for me to optimize a bioink that was feasible and that was capable of printing a particularly good construct or a tissue construct. So after that, what I did is after printing the or achieving the ideal bioink to print the construct, I had an opportunity to actually go to the Kennedy Space Center here. That's the uh, one of the places for NASA, and where most of the SpaceX launches are currently happening, which is really interesting. So we I got a grant with my professor, which is called the Florida NASA Space Grant. So with this grant, it actually helped me to go to Kennedy Space Center. And what we want to do is, since there hasn't been much study that has been done with respect to any organ systems in astronauts with respect to the effects of microgravity or radiation. So what I did was the 3D bioprinted constructs that I had, I took them over to the microgravity simulator and exposed them to microgravity effects and for 24 hours. And then I got it back and I did a couple of characterization studies on these constructs to analyze and see what microgravity effects are on human cells. So to go over some kind of background for the bioprinter part. So our main goal was for it to have its own sterile bioprinter so that we wouldn't put it inside the laminar flow hood, and to make sure that it printed well and we got to print what we wanted. So we uh, checked on and managed to make sure like our printing parameters were able to help us in what we wanted to print. And so we used different concentrations or different kinds of bioink to test this particular printability or do a printability analysis. So here on the left, you can see the bioprinter that we have. Uh, so you can see, like how I said, it has its own encased hood, so you don't have to put it in the LAF. It has it has the HEPA filter right at the back. I'm not sure how much guys can see, but yeah. And it has its own temperature control, so you can control it to whatever temperature you want it. And it has a circulation blower which helps to keep the humidity at bay. And right side, you can see a table of the printing parameters that we used for my study. So the process of bioprinting to what I did, I know there are different ways that everyone actually uses to print. So this is one of the ways that I use to make or print my bioprinted constructs. So what I did is used sodium alginate and gelatin in DI water and PBS. Then I mixed it with cells and loaded it in the syringe. And the system we use for bioprinting is an extrusion system. So using a pneumatic pump, we extruded or printed cell-based constructs. So the results from this with respect to the printer, so the couple of assays what I did was, one of the main things was live dead. So live dead assay basically gives you an idea of how the viability of the cells, so how much has survived and how much have died during this whole process of printing. So we actually maintain the constructs for about multiple days, but right here I just show I'm just showing you for 4 days. So what I did was, first we optimized the bioink with different concentrations of alginate and gelatin. So the ink I have been using for all my studies was a hydrogel based ink, which consisted of alginate and gelatin. And I had added fibrinogen because fibrinogen helps the cells in attaching to the matrix and to cause it to align along the fibrin, which I will show you images of later on. And then it helps them to actually elongate and replicate and survive. So when I conducted this particular analysis, we found that the cells would actually survive more than a day. It went on for like 4 days, as given in the graph on the right side. And then we did multiple studies for like different days. We have gone up to
Likhita?
2 days. Yes,
Yeah, voice was not audible that time. Now it is clear.
now it's okay.
Now it is better. Yeah.
Okay. So we actually did this study for almost 10 days, and we got a really good viability. I will show you the images for the different studies once you're going through the slides. So next, one of the main things to characterize a bioink is to test its viscosity. Because if you have the right viscosity, is when you'll actually get a print that can maintain its construct, that can maintain its shape. Otherwise, it's gonna just free flow, and you're not gonna have what you're gonna print. So one of the main things about any bioink that you're gonna analyze is its viscosity. So what we did is we did rheological analysis. So that is what it's called when you're trying to analyze the bioink for viscosity. And we found with different concentrations of bioink, they had different viscosities. So here what I did is I tested one particular concentration of bioink with cells, without cells, and before extrusion and after extrusion, basically to see how the viscosity of the print would change and the viscosity of the bioink would change. So what we finally analyzed from this is that decrease in the viscosity would increase in the shear stress, and shear stress is one of the main factors with respect to bioinks, because when you add cells, you want the shear stress to be minimal so that your cells survive when it's extruding out from the printer. So on the bottom you can see the Journal of Medical Devices. That's where this particular paper has been published. If any of you need any details, you can Google it and you will find it. So next thing what we did on the constructs that we printed were conduct mechanical testing to see how they could hold their like shape and structure for a couple of days. So what, and this study was conducted between to see the difference between casting and printing. So we wanted to evaluate which technique would be better. So if the casted was like equivalent to printing, then we just wanted to see why the whole of the bioprinting was such a big deal. So what we did is we did a study with respect to mechanical testing, where we have a machine called an Instron, which actually does compression testing on any kind of material, or it does tensile testing on any kind of material. So what results we got from this particular test, that was that the casted and the printed had the same amount of structural stability and construct, and it held its construct really well. So for the casted, since it doesn't go through much of shear stress or with respect to extrusion pressure, the viability of the cells will remain greater when compared to printing. But the nice part of it was there wasn't much difference with respect to like casting and extrusion printing. And then we did, so when you work with hydrogels, another work that is mainly done is with respect to swelling and degradation. So it is always good to have a material or use a material with respect to bioprinting where it can degrade within time. because you don't want to have something that you can implant in a body and not have it degrade over time. And so what we did, and with respect to swelling as well, you don't want the constructs that you've printed to swell a lot because it's going to change its shape, and probably the application of what you're going to use it for is going to vary a whole lot of what you actually expect it to. So from the results, what we got, we saw that the swelling of the casted to the printed actually showed much better results with respect to the casted constructs. And with respect to degradation, so we did a study with respect to fibrinogen and without to see the effects of fibrin, whether it really made a difference with respect to the construct or the cells. And it actually did. So like I said, it actually helped the cells to attach even better and replicate much more than what it could without having the component of fibrin in it. And what we want to see is whether it would degrade over time. So that's exactly what we saw with when you can see the graph on your right hand side, which shows the effects from day 2 to day 5. So next, so our main goal was to try and mimic a cardiovascular tissue, eventually to study the effects of microgravity. So doing some research, myself and one of my undergrads who was actually working with me and helping me out, we kind of looked up on this process called sacrificial bioprinting. So this process is one of the techniques that's being used where you can use it to actually achieve this whole or mimic the actual tissue, where you can actually introduce vascularization and make it like evolve it into an actual tissue construct. So how this particular process is done, or how what I used like the way or technique of what I use this process is for, what I did was basically print like a base structure of the alginate and gelatin, the bioink that I had already characterized, and we printed pluronic F127. So pluronic is a biomaterial that is actually used vastly right now. It's not typically good for the cells, but it's a very unique material that is actually different from any, like, for example, with respect to gelatin. So the way gelatin works, I'm sure all of you know that when you freeze gelatin, it becomes solid, and when it's in like room temperature, it's in liquid format. But pluronic F127 or any kind of pluronic, it has different variants, is different. So when you put it in the cold temperature, pluronic actually stays liquid. And when it's in the room temperature, it actually solidifies. So, which is actually a good uh, prospect, because we wanted a biomaterial that would actually have this solid feature when we print in room temperature, so as to make these vascular channels that you can see in the flowchart that is over there. So, and finally, what we did is we achieved a construct that you can see on the bottom left with pluronic and the alginate gelatin and fibrin matrix. So what we did next was we seeded cells, that is the human endothelial cells, into these channels that we made. And again, I did a live dead assay, which showed the viability of the cells with respect to different days. So I have an image here with day 4 and day 7. That is actually showing a picture of each channel that I seeded the cells in. And the viability remained pretty much the same when compared to 2 different days. So that was the first one. So coming to the second study of mine was to optimize different concentrations of this bioink. So what I did was, my main objective was to come up with a bioink concentration that would actually support the cellular viability, as well as hold its shape and construct and not just disintegrate, like lose its shape after printing. So what I did is I worked with 4 different concentrations of bioink, like you can see there, and with respect to alginate and gelatin. So when I worked with these 4 bioinks, so I conducted different analysis. So first one was printability analysis. So I used 4 different concentrations, and I checked for different printabilities, where I tested for its void area to see whether it actually held its structure after printing, or it would just like disintegrate as soon as I printed it. Like you can see a difference between the 5% 2:3, and the difference between the 10% 1:9. So from this particular experiment, what I concluded that was when you have higher amounts of gelatin, it actually held its structure better than having higher amounts of alginate. So next, what I did is tested for viscosity of all the four bioinks, and it showed that the 10% 1:9 was the most ideal compared to the rest of the concentrations of bioink that I had. So next I did structural analysis on these different constructs that I had. So how I did this is I used a scanning electron microscope to analyze the surface morphology of these constructs of different concentrations. So my main thing is to, while doing this, was to see which kind of construct had more porosity, because porosity promotes cell attachment and cell replication better than if it didn't have any kind of porosity. So from here, so what I meant by saying porosity is if you see the image of 10% 1:9 on day 7, the G, you can see the small minute pores when compared to the others, the E, F, and H, where you can't actually see those small pores that you can see in that particular image. So this actually showed that 10% 1:9 that had the higher gelatin was the ideal one to use as a bioink. So again, I did mechanical characterization on these for the day 0 and day 7. And these results also showed that the higher the gelatin, the construct was much better. But on day 1, yeah, the constructs with like alginate are better because alginate is supposed to act as a better material when while coming to viscosity. But it depends on the cross-linking technique that you use for alginate as well, and as gelatin as well. So next what I did is to check for viability, like I was talking before. So for the various concentrations of buying that I had, for the 10% 1:9, as you can see on the image and on the quantification graph on the right, it showed the least cell death. So basically that particular bioing was better than the others, and it so happened that that could actually sustain much better shear stress when compared to other concentrations of bioing. So next, I did an assay with respect to fibrin to see how fibrin actually helped. So we did mechanical testing to see if fibrin actually helped hold its structure, but I hadn't done anything with respect to like the cells. So what I did is the first two images you can see, A and B, is called DAPI phalloidin staining. So what DAPI and phalloidin staining does is DAPI is a stain that stains the nucleus of the cells, and phalloidin stains the cytoskeleton of the cells. So you can see in the pictures A and B where you can see blue and green. Basically, blue is the nucleus and green is the cytoskeleton. So you can see round cells which are present in the construct. And in figure A, the blue lines that you can see is actually the fibrin line. So if you look at it closer, you can see that the cells are actually aligning on this line. So they need some kind of ECM matrix for it to actually grow into and replicate. So what they prefer fibrin for themselves to actually hold on to and replicate and grow. And to confirm this, I did a staining called Mason's trichome, which actually has a series of steps which involves different kinds of staining, and this therefore stains different parts of the actual tissue, like for example like collagen and cells, etc. So what I did is like stain the nucleus. It basically shows the staining of the nucleus of the cells and the fibrin structures, which you can see, which are shown on the image as dark purple. So next, now coming to the major study of mine is with to test the effects of microgravity. So like I already mentioned, how the exposure to space actually causes some kind of shear stress on cells with respect to microgravity and radiation combined. So radiation, so my main thing was to test for microgravity. So what I did is after printing, I took it over to the Kennedy Space Center where they actually have this machine called a gravite, where they actually conduct different kinds of experiments with respect to, right now, the main study what they're doing is with respect to plants to see how they can do a to see how they can self-sustain in actually the ISS, the International Space Station. So most of their study actually involves plants, but none of them have actually currently seen anything with respect to human cells because it's just right now like currently being looked into. So what I did is I took my constructs that actually had human endothelial cells and used this gravite machine that is over there to actually expose or create this whole microgravity effect for 24 hours. So what I did is I tested both 2D, 3D grid constructs as well as sacrificial constructs and co-culture constructs. So the 2D was I had endothelial cells that were seated on a petri dish, and I exposed them to microgravity on this gravite system. The second was I printed grid constructs and I had cells embedded in them and I exposed them to microgravity. The third condition was where I printed the sacrificial constructs and seeded the endothelial cells in these channels and tested for microgravity. The last one was to do a co-culture model where I had endothelial cells in the channels and fibroblasts, like human dermal fibroblasts, in the whole bulk of the construct. So the results from this particular test showed that the control sample, which was not exposed to microgravity, had much more viable cells in it than the construct that was exposed to microgravity. So before I actually go into much detail on my results, so if you actually Google and check out the references that are there currently with respect to the effects of microgravity or radiation, what it actually tells you is that they have actually seen a proliferation in the cells instead of it actually dying. But what all the studies that they have done is on 2D. So the main thing or the main important thing on my study was that we were testing 3D constructs, which makes a big difference when compared to 2D. So the two main things that I tested was nitric oxide and the reactive oxygen species, which are two main factors that are found with respect to cells in the body. So it showed that the nitric oxide levels when the constructs of the cells were exposed to microgravity reduced vastly when compared to the control. And the ROS has exactly the opposite effect where the control had uh, the actual amount of reactive oxygen species that is supposed to be there in your body compared to the microgravity sample, which increased drastically. So this results were just respect to grid constructs where the HUX or the human endothelial cells were embedded in the bioink and printed as grid constructs. So this is to give you an idea of the live dead to see how the cells survived in each channel. And this was the results that I got with respect to microgravity, with respect to the sacrificial constructs, where I had seeded the human endothelial cells in the channels. So they showed the similar results what I got for grid constructs. So this also showed the same where the control with respect to nitric oxide had greater amount than the microgravity samples. And with ROS, it was exactly this opposite of what you see with nitric oxide. So the main conclusion of this study showed that the exposure to microgravity actually had some kind of stress or induced some kind of stress or cell damage to the cells, which in turn caused this particular effect of where the production of nitric oxide decreased and there was an increase in reactive oxygen species. So the current work, or currently I'm actually trying to finish up with, is a co-culture model where I told you about, where I have HUX seeded in the channel and I have the fibroblasts that are there in the bulk of the channel. So I have been doing current work on this where for the past couple of weeks where I've taken it back to the Kennedy Space Center and exposed it. So I need to do a couple of experiments with regarding to this, and then uh, I will add it on my paper that I'm about to publish, so you guys can check the results out. So I have another student of mine, a master's student, who's actually going to continue with this research since I'm done with my PhD now. So what we're going to do is basically test or do different immunofluorescence staining to test for different cell functions, and do PCR again to see the gene expression of these cells when exposed to microgravity before and after. And yeah, so I would like to acknowledge all my professors who helped me and at the Kennedy Space Center. It was a really good experience that I really had when I was there. So yeah, thank you. If you guys have any questions, let me know.
Thank you very much, Likhita. It was very, very interesting. It was
Thank you.
the perfect, you know, tissue engineering topic that you have enlightened all of us. So, so well planned and well executed studies, right? Can we have some questions from the students? Questions from students?
You can ask anything. It doesn't have to be with respect to this.
Yeah. Students don't have questions, or I'm not audible. Yeah, let them think their questions before they come up with their questions. I have a couple of questions.
Yes, let me
Yeah.
know.
Yeah, you told that gravitas, if I'm not wrong, that's the machine that you have used to check the microgravity effect on the
Mm-hmm.
3D constructs.
Mm-hmm.
There in the center, you told they're working on plants mainly with that instrument,
With that
right?
machine, yeah.
Yeah. Why they're looking plants there? They're trying to grow the plants in space, something like that.
Yeah, so the thing is, every time when the astronauts go up, they need to take resources for food and things. So, like, they run out of it easily. So, depending on how much they eat every day, so they have to keep sending different rockets again to, like, basically sustain them over there to send them extra food and material to sustain life. So their main thing right now was to see if they can grow plants in the International Space Station so that it would help them survive without doing this extra work of sending them food every now and then.
Absolutely. Yeah, absolutely. Even I have read some short article regarding such things happening in NASA. And
Okay.
one more question that I had was, so
Mm-hmm.
this, as you have proven through your research that microgravity has got not so positive effect on the 3D cell cultures, right?
Mm-hmm.
The
Yeah.
same way, what would be the gravitational condition do you think is required by these cells to grow into a three-dimensional tissue? Is it something like the zero gravity has got more effect?
So the thing is, when so the microgravity has or radiation both combined actually has some kind of effect that causes this endothelial cellular damage to these cells, so they don't survive, or the surviving rate of these cells when exposed to microgravity is pretty less when compared to the actual G on Earth, because we have been evolved on Earth, so our systems and our cells and our body are just are growing with respect to what is there on Earth. So when you expose it
Yeah,
to something that is not, then it causes some kind of stress that causes these cells to just die.
yeah. I've also read somewhere that absolutely, if we want to grow a tissue, how
Mm-hmm.
exactly it is developing inside a womb, for example.
Mm-hmm.
Womb is believed to be almost a zero gravity condition, right? So
Yeah.
something like that is really required, and
Mm-hmm.
plus this tissue engineering and bioprinting is somewhere the hope to get the tissues or the organs in the laboratory.
Yeah, it kind of gives us an insight, like you can make out more, I think, effects before anything you can go forward with. For example, even like the whole child developing, it might give you an insight of any kind of effects that it might have inside the womb or anything like that.
Yeah, very interesting. Students, please
You're on mute, Rashmi ma'am.
Students, you can ask the questions. If you have any questions related to higher studies or related to tissue engineering, 3D bioprinting,
Or if you
that was
have
very
questions with
interesting.
regarding to biotechnology,
Yeah,
it's a really interesting field.
yeah. And also that bioprinter that you have developed, that was
Mm-hmm.
very, very interesting.
Yeah, actually, now after we did it, so this bioprinter was developed in like 2018, I want to say. So after that, there are
Mhm.
companies here who are actually trying to achieve the same thing.
Okay.
Yeah. Because it's
Good.
actually a hassle to have a printer and put it inside the LAF for you to work and maintain sterility. You don't know how much
That's true.
of sterility you can maintain. So,
Yeah, that's true. That was very interesting. Yeah. Questions? Shivam, you don't have any questions?
Ma'am, honestly speaking, I don't have any questions related to this topic because it's very new for me and
Okay.
it was
Then
like
you should
quite
have more
informative.
questions.
Ma'am, it was quite informative, ma'am, but yeah, I'm not aware of those terms and so. But I have one question related to the scholarships in USA, ma'am. Like,
yes. Yes,
or
Shivam.
like people, my one of my friend, he is studying his masters and he is doing in Europe, and one of my another friend, he is doing his masters in USA. And
Mm-hmm.
talking about the scholarships, it's pretty hard to get the scholarships in USA, it seems. What is the overall scenario and picture of scholarships in USA if someone wants to go for their masters, and is there any opportunity to apply directly for PhD or something like that?
Yeah, so actually, Shivam, you can apply for a direct PhD instead of a master's. That is something you can actually do, but I'm not sure if every university has that particular system where you can apply for direct PhD. But my university does. So I have a couple of friends who are actually not from India, but like Iran, who are actually into like direct PhD instead of getting a master's. And they also have something called a fast track master's program here for students who come here for undergrad also, so that they can finish a master's program along with their undergrad. So I'm sure there are so many different things like that. And to with respect to like scholarship, yeah, it's always so what I've seen with my personal experience is when you get into any university or when you apply for any university, make sure to go on their webpage. And here, most of the universities have a lot of information with respect to its professors and their respective research that they're doing and their grants and their publications. You can find everything about them on the university website. So go over it, make like look into the what research they're doing and what you are interested in, and contact them. Trust me, they all are really, really nice. Contact them, email them, tell them if you're interested, and when you get into the university, and they will definitely do something to get you an assistantship. And maintaining it and keeping it is up to you later on, though.
Okay, okay,
Yeah.
okay. And ma'am, as a student, like, what are the skills we should acquire, like, in order to apply for masters and PhD? Like, what are the skills related to getting? It will help us in getting scholarships and all.
So what I've seen for skills to get scholarship, I don't think they'll actually see anything, but just make sure that you have some kind of research background if you want to get into the whole research field. Even especially for higher studies, main thing is if they also look at what kind of research you've done. Because if you're just involved in just getting a degree with like basically just going to class and writing exams, that doesn't show much of what you've personally achieved in your life. So here what mostly I know, it's a very different scenario when compared to India and anywhere else, because in India I know while I was also doing my undergrad, most of it when it comes to like companies and like doing these interviews on campus recruiting is all basically depends on what you studied and how good you are at your studies. But here all they see is what experience you have gained from after like attending classes and what you've done with respect to learning these things. So I think getting some kind of research like in our university in MVIT itself. So I actually worked for a semester in Dr. Nayak's lab actually. So I was actually helping his one of his PhD students with respect to one of the projects that she was conducting. So if you guys have an opportunity with respect to any of the professors in our department, go ahead and take the opportunity, and so that you have some kind of experience that you can put on your resume, which actually really matters a lot over here.
Okay, thank you.
You're welcome.
Ma'am, I have a question. Ma'am, a
Yes.
question from one student to another.
Yeah.
You have been studying, you have a lot of study years under your wing, ma'am. You have like studied for a long time.
Mm-hmm.
How do you keep yourself motivated to get through all this?
So, so actually, I shouldn't be saying this, but yeah. So when I was actually doing my master's and PhD, it's, you can't say, it is actually a really a wild ride, because you actually learn a lot. It's just, so doing a PhD is just like going for a job, but the best part is it is something that you want to do. And if you like, getting a master's is a whole nother thing, but getting a PhD is, you really need to be like motivated and involved in what you're doing. Otherwise it's really hard to get through it. So I mean, what kept me going is the whole fact about what I was working on. Because if I don't have the motivation, or if I don't like what I'm doing, then I don't think I'll go ahead, or and go ahead and do it. So my whole motivation came from the fact of what work I was actually working on. So it really helped me out.
Okay, thank you, ma'am.
You're welcome.
Anything else?
Ma'am,
Yeah.
Yes.
you told that you have made a different kind of printer, bio printer.
Yeah.
Are you trying to commercialize that or something like
So
produce it in large
I can,
quantities or it's just
yeah, so that was actually something that was made in the university. So I just use that for my research, and they are going to use that over here, but I don't think we're going to do anything, any like release it as a product that anybody can buy, because there are a lot of other different companies here that are actually selling bioprinters of different kinds. So they are actually coming up with one of the same ones now. So I don't know if it's in the market still, but yeah. You can actually Google these different bioprinting companies and you'll know.
thank you, ma'am.
You're welcome.
Any other questions?
Likita, this is Halima. How
Hello,
are
ma'am.
you?
Yeah, I'm good.
Nice work. And what about your postdoc? Are you going ahead or?
Yeah, actually, I have a fellowship lined up in Mayo Clinic. So
Okay.
I'll be starting probably the end of this month or next month, depending on my work authorization. So yeah, it is actually interesting. I was actually really happy that I got something in my own field. So the person over there at Mayo Clinic is a cardiovascular surgeon. So his research is on cardiovascular tissue engineering. So he's trying to so he works with xenografts. So he's trying to make a heart, probably like develop a heart organ and replace it in humans. So yeah.
Nice. All the best and do well.
Thank you. Thank you.
Your work is really great.
Thank you, Hariva ma'am.
Then are you applying this anywhere actually, your studies?
Applying as in?
See, a lot of things we hear like there are 3D printed organs and all that.
Mhm.
So anywhere you are trying to apply this concept?
So my whole application was with respect to seeing the effects of microgravity and radiation. So I haven't looked into like actually applying this into organs or any kind of organ transplant or tissue related conditions. So there are people working on actually. You'll be surprised to see there are a lot of people actually working on 3D bioprinting right now, because it's current hot topic that is going on. So they are all, so main things, I think they start off with like scratch or small things with respect to like skin because that is something that you can easily achieve by printing and by seeding of cells. Also, the whole cell factor is a main concern because each cell in the human body has its own mind, and it's really hard to have them to like replicate and survive. So they start from scratch and then go on to complicated things.
Okay, okay, good, good. Keep going.
Thank you.
Very nice. Mayo Clinic, even Anu Sridhar, you're from the
Yeah.
same batch or?
Yes, ma'am. Same back.
Yeah, she is also there at Mayo Clinic, no?
Yeah, she got into entrepreneurship because she wasn't into
Yeah,
research. So yeah,
yeah,
yeah.
yeah, true. Good, fine. Any other questions from students? If we don't have any more questions, Nivedita ma'am, you want to say hi to Likhita?
Hi, Likhita.
Hello, ma'am. How are you?
Yeah, I'm fine. How are you?
I'm doing good. Thank you.
It's a very good work.
Thank you.
Nice
So
to
nice.
see you in this position.
Yeah.
Congratulations and all the best for your future, Rasan.
Thank you, ma'am. Thank you so much.
So this is one of the advantage we should say to Corona, so that we can meet all our alumni who are there across the globe. We can
True.
meet them in this platform and at least get to see what they're doing, and we can interact with them.
True. That's true. That's true. But it's hard to find alumni who are still in the same field because, yeah, it's actually very difficult because there are so many different things that you can get sidetracked with. And this is
But
something new, right?
yeah, fortunately, with us, though, we could get almost some 60 to 70 in the past 1 year.
That's nice.
Almost 70 plus, I should say. 70 plus alumni were from the 2003 batch to the very recent
Mm-hmm.
batch. They are there in very, very nice places, very good work they have done, and we could get them on this platform. And we're trying to interact, connect them to our students, and motivate them in the possible way. It was really good.
True. I totally agree. That's what I keep telling everybody. Like, I wish I had someone to tell me that about this field because when we started it was just like new and we had
Yes.
no kind of exposure, right, or anything
Yeah.
with respect to this field. Yeah.
Yeah. So if we don't have any questions from the students, let's all join hands and thank Likhita for this very enlightening and very interesting webinar that she has presented to all of us. Thank you so much, Likhita, for your time and connecting back, right?
Thank you.
So
It was
once things are fine, once you fly back to India, please do visit the department.
yes, definitely.
Yeah. All the best for your future and the new job.
Thank you.
Keep in touch. Stay safe. Thank you.
Thank you. Wish you all the same.
Thank you. Thank you, all the students. Thank you, Ian, ma'am. Thank you, Arich, ma'am. Thank you all very
Thank you,
much.
ma'am.
Yeah.
Thank you. Thanks.
Yeah,
Yeah.
thank you. Thank you and bye.
Bye bye. Thank you.
Bye.
Bye.